So now I'd like to invite our three speakers to come and sit up at the front and have our question and discussion time.
This is for Dr. Griebel. When you saw the changes in the increase in TNF over time in the ones that died versus survived, did you look at changes in the PBMC populations? Is the TNF due just to expression, or is it possibly due to changes in the population?
And thank you. That's a very good question. And so I showed you the response for PBMCs. What we actually did is repeat those experiments with purified monocytes, so we could eliminate the possibility that what we were looking at, as you suggested, there might be changes in population kinetics. So we would just have perhaps more monocytes that would be present in the blood, able to respond to LPS. So if we use just purified monocytes, we get exactly the same results, showing that it's actually the function within individual cells that is altered.
Dr. Griebel, I was wondering, did you do anything, I know it's very difficult to do, but did you do anything to try to quantify the amount of stress that those animals were under? It's an extraordinarily challenging endeavor, but I was wondering if there's kind of a dose response at which point they decompensate, and that's why you see an increased response to just one kind of stress.
Okay, yeah, it is extremely difficult to quantify that. So we measured serum cortisol, which is one of the tools available now to look at that. And what we actually saw is very similar. So when we do transport, whether it's with abrupt weaned calves or calves preconditioned at the ranch, we see elevated cortisol levels at similar level, or cortisol elevated to similar levels, and then the cortisol comes back to baseline within 48 hours. So that's really the only quantitative measure. And I think that's a little bit why I ended the talk with where we're going, that we would like to have some quantitative measures of stress responses, why we're doing the genome work to see if we can identify specific signaling pathways and maybe proteins involved in those responses so that we could start to look at that. And I think for the industry, that's critical that we would have some way to actually quantify stress responses, or to say if we altered a certain management procedure, have we actually reduced the level of stress in those animals? And we will have to engage in that discussion with society as they, I think it's a number of animal behavioralists who like to say that we're licensed to produce food or animals for food by society itself. And so we have to engage with them and provide them with some measures to demonstrate that as we change our management systems, that those are actually better for the animals. So I think that's a critical question, and it's a real deficit in terms of our tools, that it's very hard to quantify stress responses.
This is also for Dr. Griebel. There was a study only a couple of years ago, I think, at University of Illinois that looked at cortisol levels using lidocaine or non-lidocaine for just budding dairy calves. And there was a significant difference, but then they compared both of those to transport, and there was a factor of 10 difference. And what my question is do you have any window or idea, is there something special about transport versus all other stressors? Because it seems to me that this has been repeated, and it seems to be related.
I can only speculate, and I have no qualms about speculating. You're right, transport and people, and that's why I say many of other groups have looked at transport, and it's always the same, that transport induces this very high level cortisol. And I don't know, I go a little bit, so restraint is a stressor that's used a lot by psychologists and used a lot in mouse models. And just taking animals and putting, like mice, in a small tube where they cannot escape. And I think perhaps Dr. Tom Notzinger could speak to this as well. But one of the things when we talk about learning how to handle animals better is always allowing them a route of escape or a way to get away from us so they can release the pressure that we put on them as people. I think when we transport animals, we put them into a confined box and leave them there for whether it's as we do for 3.5 hours or people do for 12 hours. Those animals have no escape and no way to resolve the situation. And from a psychologist's point of view, the worst thing about stress is when an individual is unable to deal with it or respond to the situation. And so that's a little bit my speculation why transport appears to be such a severe stressor.
Phil, I've got a question and comment in regards to that, and that is, so with transport, the other thing you have, though, you have dehydration, you have the fact that these animals aren't, nutritionally. So there's to me, plus what you just talked about in terms of psychological. The other thing, though, that comes into play is in terms of ambient temperature. So in terms of heat stress and cold stress, and I was just wondering if you could just comment on. First of all, you're transporting these in Canada in terms of not dealing with quite as much heat stress as we might in the time of year that you're looking at. So just curious in terms of ambient temperature when you're doing these transport studies, if it was comparable.
Okay. Yes, Chris, you're right. And especially, we're doing that's why I made a point of mentioning, we're only doing a 3.5-hour transport, which is nothing compared to animals that may be put in semis and transported for 12 or 14 hours, where these issues you're talking about, nutritional deprivation and dehydration, may be very important. And you're right, we're transporting these calves in November. They're probably warm for the first time in a month when we're transporting them, because we're usually transporting, I'm in centigrade, minus 10 or 20 when we're transporting. So I don't think we have a heat stress. And also, we're transporting 20 or 30 animals. Again, it's not like a pot where you may have 50 to 80 or 80 of these 500-pound calves there. So our conditions, that's why I think a lot of what we observe are minimal effects of stress compared to what really happens in the industry.
Knowing what you know about the effects of transport and weaning and how that affects the immune system, if you were a consulting feedlot vet and you knew you were getting high-risk, recently weaned, transported calves into your feedlot, when would you recommend to your client that they vaccinate them? How long after arrival? Okay, well, one, I'm fortunate that Fort Dodge actually funded a study to answer that question. And two, I'm guilty of not publishing it. Okay. So, in fact, we took our model and we asked that specific question, whether it would significantly impact the onset and duration of immunity to a modified live viral vaccine if, in our model system, if we vaccinated calves on arrival. So it did not, so the stressors in our abrupt weaning and transport did not significantly impact the immune responses to a modified live viral vaccine. I think the comment earlier today that we're using the vaccines at exactly the wrong time, if we're vaccinating on arrival, I would agree entirely that it's way too late, and so that's a whole other part of my research where I think getting immunity in place prior to weaning is way more important, and that was alluded to today where, that's much easier where people retain ownership. They have a vested interest in doing that. So as a veterinarian, you're faced with the question you really asked me, whether or not I'm a good politician. I didn't really answer your question. I answered the one I wanted to. But to answer your question, I think there's more important things to do, and the previous group addressed that issue. Rather than vaccinating, there's much more important things to do with those animals in terms of, that's why I wanted to show that work about the nose paddles and altering that really, I wanted to bring the message today that we can actually have effective procedures to modify stress responses, and we can do that quite rapidly. So I think a little bit some of the things Dr. Tom Knopfsinger talked about, where getting those cattle settled on feed and water, looking after the nutritional aspects and making sure those animals are sufficiently comfortable with their environment and the people handling them, that they'll show you that they're sick, I would agree is probably more important than rushing in and putting in a vaccine. So thank you for asking me that question, and I've just lost my funding from, I won't even name the pharmaceutical company.
Okay. I want to take the stress off our middle speaker and talk to the geneticist for a moment. Don, what would your estimate be using some of Allison's heritability on how fast we could make some of these genetic changes in beef cattle?
Thanks for the question. It felt a bit like Groundhog Day there. Last time I talked to vets, I got no question. At least I got one this time. Yeah, it's a difficult. Probably wish I didn't get it, because it's a difficult question to answer, because it's all driven by what's the relationship between BRD and the other traits in the economic index that Alison alluded to, and we don't know that. So Alison talked about you need large data sets to estimate heritabilities and genomic predictions, but you actually need larger data sets to precisely estimate genetic correlations. I showed you the data from Clay Center. The standard errors were around 0.2. And if you get a standard error around 0.2 around a correlation that varies from -1 to +1, you may as well just be flipping a coin or throwing a dart at it. We don't know. So that's what's going to drive it, is what is the correlation between BRD susceptibility and terminal traits like growth rate in dairy cattle, milk yield, and fertility. So, sorry, I can't answer it.
In that regard, let's turn to Allison. And it's been quite clear to me for a number of years that our number one economic trait in really relating to growth is morbidity and mortality in the feed yards due to BRD. This is probably my number one target. This definitive term, phenotype of death is definitely an economic impairment to growth in the cattle that I buy in my feed yards. And then just one other thought. I think we need to disabuse ourselves of the idea that BRD is only a feed yard disease. Most of the stockers in our part of the country the last couple of years have experienced more death loss than I have in my feed yards during the same period, of which was a great benefit for me because they died on their place before they came to mine. So I guess the question is, there's not much of a question there, but if you'd like to comment, it'd be great.
Actually, I'll touch real quick on the stress question here, because I was kind of interested to hear that what really stresses people or mice is when you restrain them and put them in a small tube where they can't escape. And that sounds a lot like flying in an aeroplane to me. And so I think we probably put Donna under, what is that, a 10-hour flight? I'm surprised you don't have BRD breaking right now. But anyway, we don't know the genetic correlation, and clearly it's an important trait, as you've alluded to. And I guess the question, and I'll put it back on you a little bit, Mike, is what sort of a premium are you prepared to pay to people that will provide you with animals where they've put the appropriate selection emphasis on BRD? Because it wouldn't surprise me if production traits have a negative correlation with health traits, because you're putting all your energy into growing, then you may be putting a little bit less into health traits. And we've seen a positive genetic trend, as you alluded to, with regard to BRD incidence. I know there's a positive genetic trend with regard to growth traits. just because they're both positive doesn't mean it's cause and effect. I don't want to make a post hoc fallacy analysis there, but I think we have seen that it's likely that that could be the case. Having said that, you can still make progress in both. So somatic cell count is negatively associated with milk production, but you can still make progress. But there has to be supply chain incentivization for people to make those selection decisions, and that's the real challenge in the beef cattle industry as a whole. I mean, I think this doesn't just relate to genetics, to be honest with you. It's the theme of what we've heard the whole day, is how do you get people to do the management and appropriate selection decisions to decrease this problem at the stockers. I totally agree. I didn't mean to. Basically, the people that make the cattle need to be thinking about the people that use the cattle's selection objectives, and how do you incentivize that, I think, is really the million-dollar question, and a really important one. So, Mike, how much are you prepared to incentivize people to make those breeding selection decisions? $20.
Yeah, on the nose clips, just an observation trying to do that is for about 2 days the calves are tremendously pissed because they're trying to suck the cow and they can't figure it out. And so I just offer, since they gained 8 kilograms in the first day, these are probably food-deprived calves. So part of your nose clip, maybe you changed the diet, you food-deprived them, then they ate a whole lot or drank a whole lot the first day. So all that works into stress, but maybe you should try a group that you put clips on for 10 days and let them get back to full feed intake to look at the lack of diet thing. The second question, do you really believe that immunity to single agents, IBR, BVD, corona, that if we select calves that are good at that, they're also going to be good against microbial pathogens and also be good against parasites? Or is there a risk if we select for BRD resistance, we open the back door to something else?
Yeah, it's a spot-on question, and it's one thing that we're looking at. We're looking at 28 different diseases in Ireland at the moment with the view to actually estimating exactly that. You have to remember that just because it happens at what we call a phenotypic level, so it happens in the field, doesn't necessarily mean that it happens at a genetic level. And if we look at the genomics, and Alison can comment on this a bit more, the major histocompatibility complex is a known causative region associated with many, many diseases having a similar trend of an effect. So there are definitely different regions of the genome that are affecting diseases differently, and it's important that if we put them all in together, we should actually get there. The biggest thing that gives me the willies about animal breeding is breeding for what we don't know we're breeding for. And we saw the great example of that in the Holstein, where we didn't monitor fertility, and we bred for milk. 20 years later, we're in a problem. We're in exactly the same issue now. Because we're not monitoring animal health, apart from maybe a bit of mastitis and a bit of lameness maybe. We're not monitoring paratuberculosis. What's actually happening to the genetic susceptibility to paratuberculosis? And we don't know.
Yeah, there was actually some interesting discussion about this at the animal science meetings a couple of weeks ago, and that is you don't want to select for animals that are super responsive immunologically, because you're going to have problems on that end as well. And so I think, as with all selection objectives and traits, there's probably an optimum there. And I guess where I would see the most potential for this will be getting rid of those 3 sires on the far right of Donna's graph that are just producing animals that are just, really getting sick all the time. And so I think that, especially in the major histocompatibility complex, the last thing you want to do is select to fixation there, because it's the variation in that complex that enables it to form antigens, right? So you don't want to be having homozygosity at that particular area. So I think that, again, there's subtleties to it, and there will be, to me, the best opportunity is to get rid of those outliers that are just throwing many sick animals, and put the appropriate selection emphasis on it, not 100% as you shouldn't for any trait.
Got one more question, and after that we'll take a 10-minute break before we start the next section.
So, Dr. Griebel, in your work, you said preconditioning in 2 weeks on the calves. Our definition in the U.S. is at least 30 days. And in the U.S., we've got lots of data to show calves that are weaned under 30 days perform, as far as BRD incidence, almost exactly the same as calves that are unweaned in the feedlot. Would you look at 4 weeks or more, well, actually 30 days or more weaning at some point and see what the difference is, because I think that would be closer to what our definition was to BRD. I was impressed that it was double, your death loss in your unweaned calves than the other. I wouldn't have expected that much either, but I think if you weaned them even longer, do you think you'd get even less death loss?
Okay, yeah. So one, we have a disease model, which is quite different from the complexities of the field situation, which is why I tried to emphasize we use a defined dose of pathogens in that. So our model doesn't recreate the complexity of the field situation. What we do know in that model, though, because we use that model to test vaccines, that even if we had we've had calves because they've come in to undergo vaccinations, so that by the time they finished 2 vaccinations, they've been in our facility sometimes for 6 to 8 weeks. We still see 40 to 50% mortality in our model. So, I mean, and that's why that model's calibrated to give that level of mortality. It was really developed for looking at protection from vaccines, and so the more we could kill, the fewer we needed in our groups. So yeah, increasing that duration to 30 days, I don't think in this model would make any difference. And the 2, I guess I'm somewhat surprised 2 weeks is adequate to bring us to a level that we see with a much longer interval in this defined challenge.
Thank you. We'll take a 10-minute break and come back.